Hello students, in this module we will learn about signal transduction by cytokine and nuclear receptors. First, let us see the introduction. Cells communicate with one another with the help of signaling molecules produced by the cells. These signaling molecules produced by the signaling cells bind to receptors present on the surface of target cells, and the recognition of the signal molecules by the receptors trigger cellular response by inducing signal transduction pathways. The extracellular signal molecules may include several hormones, neurotransmitters, cytokines, growth factors, cell recognition molecules, nutrients or waste products. These signal molecules are recognized by cell surface receptors, also called membrane receptors, transmembrane receptors, which are specialized integral membrane proteins. The binding of the signal molecules with the receptor membrane proteins induce changes in the activity or metabolism of a cell by a process of cellular communication referred to as signal transduction. Let us now see the components of cell signaling. The cellular communication signaling requires four important components. The first one is an extracellular signaling molecule produced by a cell and capable of traveling to neighboring cells. The second one is cell surface receptor proteins, which recognize and binds to the signaling molecule and capable of communicating inward into the cell. The third one are intracellular signaling proteins, which are activated by binding of the signal molecule to the receptor protein and causes initiation of the signaling cascade. And finally, the fourth one are the target proteins, which are altered upon activation of a signaling pathway and causes changes in the cell like activating gene expression. Next, let us take up the types of cell signaling. The signaling within, between and among cells may be subdivided into 5 types, that is intracrine, autocrine, juxtacrine, paracrine and endocrine signaling. First, let us take up intracrine signaling. In this type of signaling, the signals are produced by the target cell and the signals act within the target cell. The second one is autocrine signaling. Signals are produced by the target cell and they affect the target cell itself after their secretion and then binding to receptors. Such autocrine signals may also target other similar cells in the surrounding. One common example of such type of signaling is in immune cells. The next is juxtacrine signaling. The signals in such type of signaling are transmitted along cell membranes through the integral protein or lipid components of the membrane and target immediately adjacent or touching cells. The next is paracrine signaling. In paracrine signaling, the signal molecules produced by the emitting cell target nearby cells, for example the neurotransmitters. The next one is endocrine signaling. The endocrine cells produce hormones that target distant cells by traveling through the blood. Let us now take up the cell surface receptors. The cell surface receptors, also called membrane receptors or transmembrane receptors, communicate extracellular signals with the interior of the cell and act as triggers of cellular communication and signal transduction. They are specialized integral membrane proteins and are broadly grouped into 3 classes based on their structure and function. They are ion channel link receptors, enzyme link receptors, and G protein coupled receptors. Let us take up the first type, that is the ion channel link receptors. They are also called ligand-gated ion channels and constitute a large family of transmembrane multipass receptor proteins which also have ion channels for anions and cations. They are commonly found in electrically active cells such as neurons, where the ion channels open and close in response to neurotransmitters and carry out rapid signaling events. For example, the acetylcholine receptor, which is linked to a cation channel. The next is tyrosine kinase link receptors. These receptors lack intrinsic catalytic activity. Binding of ligand stimulates formation of a dimeric receptor, which then activates one or more cytosolic protein, that is tyrosine kinases. Examples of such receptors include the receptors for many cytokines, the interferons, and human growth factors. These receptors are sometimes referred to as the cytokine receptor superfamily. The next are the receptors with intrinsic enzymatic activity. These are single-pass transmembrane receptor proteins constituted by the enzyme itself and have intrinsic catalytic activity. Ligand binding activates the receptors which catalyze conversion of GTP to cGMP or removal of phosphate groups from phosphotyrosine residues. Examples include the receptors for insulin and many growth factors. The next are the G protein coupled receptors. These are integral membrane proteins constituted by 7 transmembrane helices, and these receptors activate a trimeric protein called G protein. Binding of ligand to these receptors leads to activation of G protein, which in turn may activate or inhibit enzymes that generate specific second messengers or may cause change in membrane potential by modulating ion channels. Examples include the receptors for epinephrine, serotonin, and glucagon. Let us now study the cell signaling molecules. Cell signaling molecules are generally represented by chemicals belonging to several classes such as lipids, phospholipids, amino acids, proteins, glycoproteins, monoamines, or gases. The signaling molecules that bind to the cell surface receptors are generally large and hydrophilic. For example, vasopressin, acetylcholine, etc. While those that enter the cells are usually small and hydrophobic. For example, thyroid hormone, glucocorticoids, retinoic acid, etc. There are 3 important classes of signaling molecules, that is, hormones, neurotransmitters, and cytokines. Let us see the first one, that is hormones. Hormones are the major signaling molecules of the endocrine system, although sometimes they may show local signaling, for example, islets of Langerhans to regulate each other's secretion, or may express only for local signaling in tissues, for example, angiotensin. The next class are the neurotransmitters. These are signaling molecules of the nervous system. They may also include neuropeptides and neuromodulators and some neurotransmitters. For example, catecholamines may be secreted into the systemic circulation by the endocrine system. Next are the cytokines. These are signaling molecules of the immune system, commonly with a paracrine or juxtacrine role. Although They may also enter the systemic circulation and cause effects including alteration of iron metabolism or body temperature, etc. Next, let us see intracellular signaling molecules. Cell signaling also involves the role of several intracellular signaling molecules, termed second messengers. For example, 3,5-cyclic AMP or cAMP, 3,5-cyclic GMP or cGMP, 1,2-diacylglycerol or DAG, inositol 1,4,5-triphosphate or IP3, and various inositol phospholipids or phosphoinositides and calcium ion. The binding of ligands to cell surface receptors leads to elevated concentration of one or more intracellular second messengers, which triggers the activity of one or more enzymes or non-enzymatic proteins. Next, let us take up cytokine receptors and signaling pathway. Cytokines are substances produced by immune cells and they have the potential to affect the activity of neighboring cells. Cytokines bind to cytokine receptors present on the cell surface with high affinity. These receptors are linked to important signaling pathways that play a role in cellular activation, survival, proliferation, and differentiation. The cytokine receptors contain 1 to 3 chains, that is alpha chain, beta chain, and gamma chains, and associate with one or more members of tyrosine kinases termed Janus kinases or JAKs. In contrast to some type of nuclear growth factor receptors, cytokine receptors are devoid of catalytic activity. Instead, the interaction of a cytokine with its receptor induces phosphorylation of the receptor through cellular tyrosine kinases such as Janus kinases or JAKs. Cytokine receptors can activate many signaling pathways like the JAK-STAT signaling pathway by means of phosphotyrosine residues. The phosphotyrosine residues are recognized by HH2 domains on the signaling molecules such as signal transducer and activator of transcription or STATs. Let us now see the mechanism of JAK-STAT signaling. Cytokine signaling plays important role in human body regulation. Cytokine signaling helps the immune system in keeping the human body healthy. When the macrophages and dendritic cells engulf foreign particles, they send cytokine signals to nearby dormant lymphocytes, which recognizes the signal and activates the lymphocytes that are specialized to recognize specific antigens. Since some cytokines do not act locally but travel long distances through the body, they are sometimes classified as hormones. However, unlike hormones, they are not secreted from glands. They are secreted from glial cells of the nervous system and are essential for embryonic development. The most common pathway for cytokine signaling is the protein kinase transduction cascade, for example, JAK-STAT signaling pathway. When a ligand, which is usually cytokines such as interferons and interleukins, binds to a cell surface receptor, it causes dimerization of the receptors and brings the receptor-associated JAKs into close proximity. The JAKs phosphorylate each other by a process called transphosphorylation. Then the JAKs add phosphate groups on tyrosine residues of the receptors and create binding sites for proteins with HH2 domains such as signal transducer and activator of transcription or STATs. The binding of the states to the phosphorylated tyrosines on the receptor is followed by their tyrosine phosphorylation by JAKs, which leads to the dissociation of the activated STATs from the receptors. The HH2 domain of each activated STAT then binds to the phosphorylated tyrosine of another STAT to form hetero or homodimers. The dimers are translocated to the cell nucleus, where they bind to the DNA and induce transcription of target genes. Next, let us take up nuclear receptors. Nuclear receptors are cellular proteins that recognize steroid and thyroid hormones and certain other signaling molecules. Signaling pathways involving nuclear receptors play a role in regulating development, homeostasis, and metabolism by controlling the expression of specific genes. They can also bind directly to DNA and regulate the expression of adjacent genes, and hence these receptors are often classified as transcription factors, which is a unique property that differentiates them from other classes of receptors. When a ligand binds to a nuclear receptors, it results in conformational change in the receptor molecule, leading to activation of the receptor, which in turn up or down regulates the expression of genes. The nuclear receptors commonly exist as monomers, homodimers, or heterodimers, and they recognize DNA sequences termed as hormone response elements or HREs. The nuclear receptor superfamily is ligand-activated transcription factors. Binding of ligands to the nuclear receptors result in transactivation of specific genes. The action of the receptor is modulated by numerous growth factors and cytokine signaling cascades that involve receptor phosphorylation or other post-translational modifications, typically within the N-terminal transactivation domain. Let us now see the structure of nuclear receptors. Nuclear receptors commonly have a structure comprising of the following components. The first one is a highly variable amino terminal domain with many transactivation regions, which is also known as the AB domain, also called activation function 1 or AF1. The second one is a conserved DNA binding domain in the center containing 2 zinc fingers, also called the C domain. The third one is a short region responsible for nuclear localization, also called the D domain. And the last one is a large, well-conserved ligand binding domain in the carboxy terminal, also called the E domain or LBD. Some nuclear receptors may also possess another highly variable carboxy terminal tail, which is called the F domain, mostly with unknown functions. Next, let us take up types of nuclear receptors. Nuclear receptors are commonly grouped into 4 subtypes on the basis of their mode of action, that is, type 1 receptors, type 2 receptors, type 3 receptors, and type 4 receptors. First, let us see the type 1 receptors. It includes receptors such as the receptors for androgen, estrogen, and progesterone. These receptors are anchored in the cytoplasm by chaperone proteins, for example, heat shock protein 90. When a ligand binds to it, the receptor becomes free from the chaperone, which allows homodimerization followed by exposure of the nuclear localization sequence and entry into the nucleus. Inside the nucleus, the ligand receptor complex activates target genes in association with transcriptional co-activators. The next one is type 2 receptors. In contrast to type 1 receptors, the type 2 receptors, for example, the thyroid hormone receptors, the retinoic acid receptor and peroxisome proliferator activated receptors, PPAR, alpha, beta and gamma. They are present as heterodimers bound to their specific DNA response elements inside the nucleus. They exert active repressive functions by interacting with co-repressor complexes in the absence of ligand. Ligand binding leads to dissociation of co-repressors and their replacement with co-activator complexes, which activate target genes by de-repression or opening up of chromatin. The next are the type 3 receptors. They function in a way similar to type 1 receptors and differs only in the organization of the hormone response elements or HRE, which is a direct repeat in type 3 receptors in contrast to the inverted repeats of type 1 receptors. The next are the type 4 receptors. Type 4 receptors bind as monomers to half-site hormone response elements. Now coming to the conclusion. So we have seen that cell signaling constitutes a complex system of cellular communication that helps in maintaining cellular activities and coordinates cell actions. The perception of signals by cell receptors and induction of signaling pathways allows cells to respond to their microenvironment and governs important processes in multicellular organisms including development, tissue repair, immunity and homeostasis. Different extracellular signals are recognized by receptor proteins, triggering intracellular pathways involving second messengers that ultimately modulate cellular metabolism, function or development. Thank you.